Hello everyone, welcome to another lecture of drug delivery principles in engineering. In the past 5 lectures we have basically gone over some of the basics of the drug delivery, why it is required, what are the different scenarios that are currently used in clinic, and what is it that we would like to achieve. Then in subsequent classes we talked about one was prodrug, and then another thing was we talked about lots of polymers, some properties of the polymers. So, all this we discussed so that we were kind of building up the base before we go into the actual drug delivery concepts that we are going to use for the rest of this course. So, now we are almost ready to essentially talk about some of the polymers that are widely used in drug delivery and how those are much better, or at least give you a lot more control for clinical scenarios, and we are going to now start going much more deeper into different kinds of mechanisms and different kinds of systems that are out there. Okay, so just a quick recap of what we did in the last class. We talked about polymer properties, essentially molecular weight, how do you calculate that, the number average molecular weight, or it could be the weight average molecular weight. We did couple of exercises on how to calculate different things if you know individual components. We also talked about what is polydispersity. Essentially it is a measure of how much dispersion is there between the different molecular chains that are present in a system. We talked about crystallinity, how crystalline the polymer is, and associated measurement of temperature with that is Tm, which is the melting temperature at which point the crystallinity is gone. And then for some polymers the crystallinity does not exist, it is only amorphousness, and essentially this is again as associated time point with that is, or associated temperature with that is Tg. And so now today we are going to talk about biomedical polymers. So far again, as I said, we talked about in general the polymers and its properties. Now we are going to discuss more into biomedical polymers. So just again quickly defining some of the terms. Biopolymers, what is biopolymers? Biopolymers are polymers that can be safely used in biological or medical application. So typically these polymers are naturally present and hence they are called biopolymers. So again, biopolymers can be divided into 2 different classes. One is synthetic biopolymers, and as the name suggests, these are synthetic, so they do not occur in nature. As I earlier said, biopolymers are something that can be used for medical applications. They may or may not exist in the nature. So in this case, the synthetic biopolymers are something that we synthesize. These are chemically synthesized polymer, does not occur in nature. They are designed specifically for a particular use of a disease. So this could include delivery, this could include tissue engineering, some prosthetics, and again we are going to talk about all these as we go along in this course. And then the other class of course is the natural polymers, which as the name suggests, these are naturally occurring. So these are derived from plants or animals or some other organisms, and these are then isolated, purified, and then they are used for different applications just like the synthetic polymers. So just before we go into this, here is a good review that you guys can essentially go through, and it's a very general review about some of the advances that are made in biomaterials for drug delivery. So just something that I would like you guys, if you want more information about this, you can go through this review, although this is not a part of this course. So again, some of the major properties and some of the major differences between synthetic and natural polymers. So synthetic polymers, these are chemically synthesized from their monomers. So some common examples are PLGA, PET, and PEG, and many other. And natural polymers are something that are derived from organisms. So these could be cellulose, chitosan, hyaluronic acid, proteins and DNA, collagen, one of the most abundant protein. Synthetic polymers, they can be, since we are designing them, we can easily tailor them to different properties. So let us say if we want the polymer to be faster degrading, we can incorporate that using the monomers which are hydrolytically cleavable at a faster pace. If we want something that is has a certain crystallinity, we can again choose polymers on the basis of that. However, natural polymers, of course, they are their native forms, so you can't really change their properties a whole lot. Again, with synthetic polymers, since we synthesize them, we can modify them depending on what again is the application. However, with the natural polymers, although modification is difficult, but then they can still be modified. So they can be conjugated to different things using some chemistries, and so the modification is feasible, although not to an extent which you can do with the synthetic polymers. And then of course, the large scale purification and production is very feasible with these synthetic polymers, just because you can make big reactors and the supply is essentially just the monomer. So, as long as you have enough monomers, you can scale it up to whatever amount. However, with natural polymers, you are kind of dependent on where to get it from. So, if it is a plant source, you do not really want to cut too many plants. Similarly, if it is derived from animals or sea organisms, you are essentially dependent on how much is the supply and how much you can extract that out from the nature. So, typically the large scale production is sort of difficult, and they are synthesized in small batches, which is another some of the shortcomings that people point out about natural polymers, because they are synthesized in short batches. So, each batch is, although there are protocols in place, but they are always treated slightly differently, and so there could be batch to batch variation with natural polymers, whereas synthetic polymers you can make a huge batch and you do not have to worry about the batch to batch variability, at least for your study. So, some of the natural polymers that are present in the biomedical applications, again there are several of them we talked about, we gave example in the last slide. So, here are some more. So, you have proteins and protein based polymers. These could be used for different applications, such as they could be absorbable, they are of course biocompatible, widely used in tissue engineering. Example of the proteins is collagen, which is basically the most abundant, one of the most abundant protein present in a body. This is a structural protein and very widely used in tissue engineering. Another is albumin. This is another protein that circulates through our blood, and again very widely used. You can have polysaccharides. So, these are essentially sugar moieties that are present in our body. So, these could be agarose, which is derived from a seaweed. This could be alginate, this could be cellulose, several of them, and all the different applications are written here. You do not really have to remember all these applications particularly. We will talk about some of these as we go along. This is just for your reference that there are a wide variety of natural polymers that exist, and we use them for biomedical applications quite a lot. So, what should be the desirable property of the biopolymer? So, one thing is certain, it should be non-immunogenic. So, of course, if it creates any kind of toxic response in the body, any kind of inflammation in the body, then that is a complete no. The patient will never feel better with those kinds of polymers. So, it should definitely be non-immunogenic. It should be non-toxic. Of course, we are trying to cure the patient. So, these polymers should be very compatible, that they do not really cause any tissue death or even the damage, a small damage to the tissue. The properties, again these depend on specific applications. So, what are the mechanical, chemical and electrical properties? Let us say if I want to put a material that is going to stabilize my bone, that is a polymer that I need to be structurally very strong. So, I want very high mechanical properties. If I want something to put for our neural implants or something related to brain, they should be able to conduct the signals. So, the electrical properties become important. So, again all of these properties are important, and in which one is more critical then depends on the application that we are looking at. And of course, as we already briefly discussed, is they should be easy to scale up. I mean it should not be like that we can only get a milligram of that, let us say in a year, something of that little quantity is not going to help. So, they should be reasonably scale up. I mean we may not be able to get quintals and tons of these materials, but then still depending on the application, if we require a certain amount, we should be easily able to get that. So, mass production should be easy. In some cases, especially in cases of drug delivery, it is desirable that the polymer does not remain for longer period. I mean essentially, let us say if we have a fever and we want a drug to be given for 5 days, this is the maximum we want the polymer to be present. So, in that case these polymers should degrade and come out from the system as well, or excreted or metabolized, any of those mechanisms. So, then the degradability of the polymer also becomes important. However, this is not essential. I mean again as I said, if you are looking for some structural polymers, something that gives you strength in your bones or something like that, you do not want it to degrade, at least not anytime soon. So, these are again application dependent. So, as this is a good segue into this slide, so how would you choose biomedical polymers? So, as we said, the major thing is what is the application. So, there are several library of these biomedical polymers out there, but the one that you choose will depend on what is your application. Then there are other things, there are what route of administration you are going to use. So, there are several ways you can administer a particular polymer in the body or a particular drug in the body. You can directly put it into the veins, you can take a tablet orally, you can put it under the skin, or you can put it on some mucosal surface like lungs and all by inhalation. And then there are several others, and we will talk about route of administration in the later part of the course. But again, you will choose different polymers depending on what you want to achieve, different sizes of them, different properties, all will depend on that. Biocompatibility is a very big term that is being kind of used in the field. However, this depends essentially on where and how it is going to interact with our body. So, biocompatibility for lung tissue might be very different from the skin tissue, which again might be very different from the brain tissue. So, this biocompatibility is essentially defined on the basis of the application itself. And then as we discussed, we may also want some kind of degradation to happen, so some kind of bioerosion to happen. So, again this again depends whether we want a permanent implant or we want it to be temporarily injected into the body and gets cleared out. And then also what are the surface properties, do we want the proteins present in the body to interact with the surface, sometimes we do not want that to happen, and again all of these we will discuss, but these become all of these are some of the properties that or some of the things that we will need to consider before we choose a biomedical polymer for our application. So, much further on that, again mechanical properties are important. So, how much load does the device needs to bear? So, again if it is a bone implant, you need it to be structurally very stable. If it is something that you are just putting under the skin for something to release out, it does not really need to bear any kind of load on it. So, the mechanical properties of those implants will be very different. Do we need a defined shape, or the shape is not very important? All of these become important in that case. Whether we want it to be environmentally sensitive, and what essentially that means is there are polymers which will respond to the environment they are in. Let us say if it is a disease environment, they may behave differently than in a healthy tissue. So, that allows us to kind of make it very disease responsive. So, only the drug will come out if there is a certain kind of a disease symptom that is present. Maybe it might be high temperature due to fever, it might be low pH at the site. So, all of that becomes important, and again all of these things we are going to go further into details as we go along in this course. Then we have permeability. So, whether we want these polymers to be permeable, things may come in and out on these polymers. Large scale production we again talked about earlier, and then whether we want them to be transparent. So, if let us say we are designing something as a eye lens or a cornea, we essentially want them to be transparent. In other applications we may not care. So, again it just essentially depends on what is the application we want, and depending on that there are several properties that we will have to consider before we choose what kind of polymer to go with. Again this is a laundry list of lots of things. I do not expect you guys to remember this, this is just for your information, and this will be present in the slides, so you can go through these. Essentially these are polymeric properties needed for specific biomedical applications. So, there are several of them listed here: dental, ocular, orthopedic, vascular, and several others. So, you can just go through that for your own interest and in free time. This is again not something that you guys should remember. Okay, so let us define some more terms. We have biocompatibility and biodegradability. So, what is biocompatibility? As we mentioned previously, it is the property of the materials, how they are interacting with the body, whether they are causing any kind of adverse reactions such as inflammation or toxicity when they are placed inside the body. So, eventually for any application we would like the biocompatibility to be high, and which essentially means that they are causing less and less of these adverse reactions. This is again very application dependent. A material may be very compatible in let us say eye, but may not be very compatible let us say in liver. So, but even then we can use the material in the eye if we want, but then it does not mean that it is completely biocompatible, it just means that it is biocompatible for the certain application. And biodegradability is essentially refers to the breakdown of the polymer into smaller units, which can either be then excreted or get absorbed into the system. This is again a very general term, and then there are several related terms that you will hear in the field. Some of them are bio-erosion, bio-absorption, bio-resorption, and we will talk about this as we go along in this course. But essentially all of them have similar meanings, although there are certain differences that exist between these terms as well. So, biocompatibility, let us talk about host reactions to the polymers. What are the different things that may go wrong, or what are the different things we need to take care about it. So, essentially this is a result of how a physiological process kind of acts on a new polymer or a new material that you put inside the body. And essentially the key here is that the material should be compatible enough so that the body can tolerate it and coexist. So, it could be biomimetic if you want to call it like that, or the body should not really consider it to be a threat to itself. So, essentially all material that you put in the body are going to interact with the body. What is the extent of this interaction is basically what is important, and not only the extent, but whether the extent is positive or negative or neutral is also very important. So, some of the key interactions when you put things in the body is, of course, there will be blood present at the site that you are going to implant it. So, the blood will interact with your material. The blood contains several proteins and platelets. So, how they interact with that surface becomes important. The blood also contains several components of complement system, which is immune response against foreign things. It's one of the immune responses that body generates. So, how those complement proteins tackle the material that you put in is important. The immune cells, leukocytes, how they are adhering, whether they get activated. Sometimes what the body does, it does not like the material and it wants to just completely wall it off, and so that is called encapsulation or scar tissue. So, what body will do is, if it can't clear it by itself, it will just surround it with lots and lots of proteins and cells, and essentially kinds of isolates it from the rest of the body. And so that is called encapsulation, or foreign body reaction is also an advanced stage of that. And it could also be in terms of infection. So, whether your material may contain something pathogenic that may infect the body. So, all of these topics will be covered in much more detail when we go to the inflammation part of this course. So, assessment of biocompatibility. So, again there are, as I said, it depends on application, and there are several ways to go about with it. The first is, before you put it in the body, you can test it with some of the cell lines, some of the cells that you may have access to. So, you can put your cells on the material, you can see whether the cells survive or they die. You can take the degradation product of these materials and expose them to cells to see what response do the cells give once they are exposed to materials from your particular biomedical polymer. You can look at the biochemical function, you can see how the cells are producing different enzymes, whether the cells can perform their normal function. Let us say if it is a bone cell, whether it can deposit calcium and mineral, things like that. Then you can obviously go in vivo, you can put it in the body, you can use some small rodent models for that, and you can then kind of do histology, which essentially means sectioning out the area where you put it and see how is the body responding to it compared to the healthy tissue itself. And so, you can do it at different time points to determine what is the extent of the reaction and how the reaction is proceeding over time. And then of course, you can get access to blood and then test the blood out on these polymers, see whether the blood is clotting on it, whether the blood is lysing on it, the blood cells are lysing on it, whether it is causing any kind of systemic toxicity. And let us say you want to use a material that is not really biocompatible, what would you do? So, there are strategies out there which will help you make it more compatible than it is, and so you can modify it with some surface. So, You can take a highly biocompatible polymer such as polyethylene glycol or hyaluronic acid and just coat it on the surface. So, what will happen is the body will only see, let us say this is a material and I have put peg chains all around it. So, now the body can only see the peg chains when any cell comes, and it feels that this is compatible and it just goes away, it does not really do anything adverse to your implant, and so that basically causes you to improve the biocompatibility of the implant that you want to use. You can again surface modify it further. So, if you let us say you want to reduce protein adsorption, so again the same strategy will be useful. You can coat it with some of these materials, and we know that the protein adsorption on these ones is low. So, in general your device will now has lower protein absorption. You can then also devise strategies where let us say you can't prevent the cells to come and attach to it, but what you can have a device that is carrying anti-inflammatory molecules in it, which then slowly get released out. So, let us say even if your immune system is coming in and interacting with it, which you in the first place did not want it, but then with these molecules coming out into the immune system, they will tell the immune system to calm down, don't act as if this is a foreign object, and that will improve the biocompatibility of your material. Or you can use some alternative routes. Let us say if you only want to treat a local disease, let us say it is a wound on the hand, maybe you do not need to inject it in the whole body, you can apply it topically. So, you can change the routes of delivery to avoid kind of systemic toxicity. And again, as I said, all of these things depend on applications, and here we are going to talk about some general strategies before we go into applications of different things. You can also combine properties to satisfy need. You can have copolymerization, as we talked about. Let us say you are initially going to use AAA polymer. So, A is the monomer and you are going to make a poly A. This poly A works very well for you for everything that you need for an application, except that maybe it is not very mechanically stable and you want the mechanical properties to be enhanced. So, what you can do, you can copolymerize it with let us say ABA, and maybe B is more structurally stable. So, the copolymer is somewhere in the middle, but it improves the mechanical properties enough so that you can use it. So, that is just one example, but you can do the same with chemical properties. All of these can be adopted to kind of improve the properties for your own application. So, as listed here, chemical and mechanical properties can be adjusted. You can even combine synthetic and natural polymers. There is no reason why you want to keep it completely synthetic or completely natural. So, if one of the property for natural polymers is better, you can use that and combine it with synthetic. In particular, you can modify hydrophobic and hydrophilic groups to attain different kind of degradability, different kind of interactions with the body, and all of that is feasible. You can blend things. So, you do not really have to copolymerize. Let us say you are going to use a big implant that is made out of A, you can just blend B in it. So, let us say this is one polymer chain, you can just blend the polymer B into this, and that will still improve the mechanical properties of whatever you are trying to achieve. Maybe you want a faster degradation, so this will degrade faster because let us say B degrades faster. So, all of that can be achieved, and this is again very widely used for drug delivery and tissue engineering. We are going to talk more about that. And then you can network things. So, mostly used in tissue engineering, what it is essentially creating a 3D polymer environment having tailored properties. So, instead of having them as separate, you can have chains of A, and then you can network this with let us say chains of B going right through them. So, that can also be achieved. Okay, so let us talk about polymers and controlled drug release. So, what are the different polymers that are used? So, of course, there are non-degradable polymers such as implants and things you use for oral delivery, because you know that these things are going to get excreted out. There are membrane controlled devices such as skin patches. So, you can just put it on the skin, let the drug come out, and then once the time period is over or your disease is cured, you can just remove the patch. So these you don't really want them to be degradable. They can stay wherever they are, and then when they are done when you're done with them, you can just remove them out. Or these can be degradable polymers. So again, this is where the most of the research is currently going on, more fancier systems. So these are something that you are going to actually inject into the body. Let's say you put it in the blood, you do not want to circulate in the blood forever. You can't really remove once you inject it into the blood, because you can't drain out the whole blood in a human or in an animal. Or let's say you put it on a mucosal system. So these are something, once you inject them, they are there unless they degrade. So you most of the time you will want them to be degradable polymers. So anything you are making micro and nanoparticles, they are too big now to remove from the body unless they break down. So you want them to be degradable. Hydrogels are another class of polymers we are going to talk about. Any degradable implants and matrix type of polymers that again will be discussed later in this course. So biodegradability, again, most drug delivery devices are typically temporary, because you are trying to cure a disease, and once the disease is cured, you do not want that device to be there anymore. So that's where the widest use of biodegradable polymers is, and let's get the terminology right, as we talked about 3-4 slides back. So biodegradation is nothing but degradation by biological molecules. This could be enzymatic, this could be microbial. Bioerosion, on the other hand, is the erosion of the polymer into the water soluble products under physiological conditions. So this could include both physical and chemical processes. So technically speaking, bioerosion is a wider term, and biodegradation is a part of it. So if it's something that is hydrolytically cleavable by water, it comes under bioerosion. It's not under biodegradation, but you will see that this field has grown enough, and there are so many papers and so many literature talking about hydrolytic degradation is also biodegradation. So just wanted to kind of introduce you to this concept. However, you will see both these terms being used very interchangeably. Another note here is a polymer that we are going to talk quite a lot about is PLGA or PLA, and that's something that is not biodegradable but bioerodible. But again, if you look into the literature, you will find that people talk about PLA being biodegradable all the time, and now it has come to the point that is very kind of being accepted that okay, bioerosion and biodegradation can be used interchangeably. However, strictly speaking, bioerosion is different from biodegradation. So there are several modes of bioerosion. One is a physical mode, which essentially could be bulk erosion. So what do you mean by bulk erosion is that the rate of water penetration into the solid device exceeds the rate at which the polymer is eroded. So what does that mean? That means that let's say I have a device, and this contains lots and lots of polymeric chains which can hydrolytically cleave in presence of water, and the water is actually free to go in. Right, so a water molecule can potentially go in throughout the polymer device. Now, if this is the case, and we are saying that these chains can be degraded by the water, what will happen is that the erosion will happen throughout the matrix, right. The water will go to all regions, and at all regions the chains will start to break down. So over time this will start getting irregular in shape, so this will become something like this after let's say few hours, and then further down it will maybe just break down into individual small units, and then they will also degrade over time, right. So most hydrophilic polymers are like that. If they are hydrophilic, of course that means that they love water, and that means that the water can go through in them because the water will also like them, and they will be bulk eroding. There could also be surface erosion, which basically means that the rate at which the water penetrates in the polymeric device is slower than the rate of erosion. So what that means is, let's say if I have a device again containing lots and lots of polymer chains, however the water molecule cannot go in at a rate which is faster than at the rate which it will degrade the outer surface. So in that case what will happen is this device is going to maintain its shape, and only the edges will degrade, and it will take this shape, which is again further going to take this shape, and so it is going to eventually go on and very systematically only from the surface it is going to keep on eroding. So, the device will become thinner and smaller over time, however, it will more or less maintain the shape. Do you guys can think of any example you see in the real life with this? So, a good example is a soap. So, if you use soap, the soap bar essentially keeps on getting thinner and smaller as you go on, it does not really disintegrates into small units. So, that is a surface erosion, because the water is not able to penetrate inside and only from the surface the soap is eroding. Whereas bulk erosion, you see any kind of basically, let us say you take a sugar molecule, the water is going to penetrate right through and it will just completely disintegrate in your mouth. Okay, so what are the different factors that influence hydrolytic bioerosion? So, you can have a backbone hydrolysis is the most common mechanism of erosion. Typically, you have a long polymer chain, and this is the backbone, and there are of course side groups to it. And then this particular long chain has some hydrolytic bond that is being attacked by the water molecule and eventually degrades them into smaller units. So, this is essentially the most common route that is used for synthetic biopolymers. The main factors that determine the erosion is the chemical stability of the backbone, of course. So, there are several functional groups that are hydrolytically cleavable, such as anhydride, esters, amides, and their rate of degradation is listed here. Anhydrides degrade faster than esters, which degrade faster than amides. And so, based on what the bond structures are there, what different functional groups are there, the degradation will eventually change. The hydrophobicity of the monomer is also important. So, let us say if the monomer is fairly hydrophobic, then the water will not try to come close to that monomer, and so that would mean that the hydrolytic degradation for that is going to be slower, just because the water cannot really access it. The morphology of the polymer is important. Again, the crystalline polymer are dense, and as we said that once you are achieving a crystallinity, it is very tightly packed structure, and even the water is not able to penetrate, the light is not able to penetrate. So, so depending on what temperature you are using, typically at higher temperatures you will always see faster degradation, however there are some exceptions. So, another point here is the PLA, which is a semi crystalline molecule, while PDLA, which is essentially the mixed isomer of PLA, is amorphous. So, which one will degrade faster? Of course, the one that is amorphous will degrade faster, just because the chains are more separated out in the PLA with the DL form compared to just the PL in the L form. And of course, the molecular weight of the chain, what is the fabrication process you have used, the geometry of the implanted device, how much surface area is there, surface to volume ratio, all of this will in fact influence your hydrolytic bioerosion. Okay, so we will stop right here for today, and we will continue with more of these biomedical polymer properties in the next class. Thank you.